Thank you, Fridbjörn, and thank you so much, Richard and Pia, for the arrangements and the Emerge group that have set this scene today. It's so nice to be back here again. It's become a tradition now, and we really enjoy it. Going to the meeting, meeting all our colleagues and all your patient representatives, but of course also sneaking over to the Red Lion now and then and have a pint, or a half a pint at least, sometimes. So Richard asked me to talk a bit about multi-omics, biomarkers, and discovery for diagnostics and disease mechanism. And you already heard beautiful talks this morning and also during the week already, so it's a bit, pretentious to actually give it so much more on that side. But I will show you a bit what we are doing and also a plan that we have just launched for an investigation that I hope will help us to get us to a robust and clinically available and clinically useful biomarker or a set of biomarkers for future studies. So just to start, I think we all are so aware of the situation we have with ME/CFS, and we also know that there is still a lack of biomarkers, a lack of FDA-approved treatments, although we now see finally that there are ongoing trials, and we hope that those will be successful and lead forward in the prospects. And one thing that we have to keep in mind, I won't dwell too much on it, is that the disease also differs over time, changes over time. Although it has its chronicity, it will change, and it will be difficult to find perfect biomarkers for every stage in the disease. And Chris Armstrong, a colleague in Melbourne, is, for instance, looking into this more temporary resolved sampling and so on, which is very important. When it comes to the symptoms, we all are aware of that. That's a multitude of symptoms, and fatigue and post-exertional malaise, of course, orthostatic intolerance, and all that we have to look for. And of course, is there common mechanisms? I think we see during the week and today also that there are common grounds for many of the symptoms actually, and maybe the biomarkers we are going to find will be connected to those, and there are some indications already pointing in that direction. I don't want to show you a case control, but I selected this guy instead. See if he is moving, yes, slightly. So here's a post-exertional malaise individual then. We just came back from a 40-kilometer trail ride, and he is, I don't know if you can hear it, he's calmly snoring, quite loudly actually, but I don't know if you hear the sound. Let's see if I can get it snoring again. You have to crank up the speaker maybe. Anyway, snoring very much, really relaxed, and also enjoying his new cleaned box, as you can see here. And this is a very nice setting of showing that this individual is very confident with you, and you can enter the box and you can do things around him. He keeps snoring and just laying there on his side. When it comes to the biomarkers then, of course, the biomarkers will represent different things during different stages of the disease development. And as you know, when you're in the normal stage, healthy stage, you still will have variability in your markers of different kinds. And of course, that's important to be able to monitor them in the healthy state, to be able to see where the break is, where things go wrong, and maybe turn into a more pathological state. And early detection is, of course, very important, because then you have a chance to actually catch the disease early and then set in a treatment that would hopefully reduce the risk of having more chronicity and permanent damages to organs or to the systems. Then, of course, prognostic biomarkers has been mentioned to be able to tell what is the progression of the disease, but also how is the individual responding on the treatment that we are then testing. What we do, we are relying very much on high-resolution mass spectrometry, and I won't go into the very details on how that technology is, what it's doing, but it's measuring all molecules that are present in a certain concentration in a biosample. It can be tissue, it can be a body fluid, it can be small, very small materials, as I will show you. And the beauty with that is that it can cover without, you don't have to know exactly what you're looking for. You can actually screen and actually measure everything you have in your sample with some sensitivity. When it comes to the proteins and proteomics, you may know that we have extremely many different proteins in the human body. We have over 1 million proteoisoforms, and there is still no technology out there that can cover all these million isoforms of a proteome. We have heard about O-link technology, for instance. Today they can claim about 5,400 proteins by a selective enriched, enhanced detection method, as Douglas presented earlier today. Very, very fine and very precise. On the other hand, you have to know what you're looking for, so that's the limitation. Proteomics, in this case, with mass spectrometry, you don't need to know what you're looking for, so you have a more screening ability. If you have the metabolome, then, of course, getting even more complex. Here we have millions of different potential substances and elements that could interact and relate to the things that happen in our lifestyle and life, epigenetic changes, aging, environmental changes, disease, drugs, nutrition, and lifestyle. So it all makes up our phenotype. And of course, measuring that with techniques like mass spectrometry, we generate enormous amounts of data. We, specifically in our group, we target absolute quantitation methods from metabolomics, just to be able to relate between different cohorts. So we have all measures in micromolar concentrations or nanomolar concentrations, depending on what analyte it is. But that can also be a sustainable information that lasts for many studies and over decades and years. Of course, bioinformatics is extremely important, and that's still one of the hurdles we have to be able to crank through all the data we generate. So it's very nice to see the presentations where this is actually now moving in the right direction for making it available for all of us. The latest development now is pushing the technology in mass spectrometry-driven analysis a bit more forward. So with the latest editions of mass spectrometry, you can run fairly high throughput analysis. In this case, I've just listed one setup that we are using with 37 plasma samples, blood samples analyzed per day. And that in reality then generates about 1,000 proteins in plasma from individual samples and reaches about 2,500 proteins in total that can be detected in a plasma sample. So still a bit lower than what we have in O-link technology, for instance. On the other hand, if we look at the peptide levels, we see that they are actually 10 times higher. So we get, in total, maybe 25,000 peptide tags instead. And that enables us to actually look at isoforms of proteins and also help us to identify post-translational modifications. And that's still not so much possible, other possibilities around there out with other technologies. The technique is now also pushed towards single-cell proteomics, and we are establishing this in our group now, so we can actually isolate single cells in circulation, for instance, or from tissue extracts, put them in wells, small wells, and then go in with a specific sample preparation technique to isolate the proteins. And what is shown now is that you can get about 6,000 proteins from a single cell analyzed with good reproducibility. When on the peptide level, about 40,000 peptides. So impressive push for sensitivity. And one can maybe wonder, how come it's easier to detect a lot of proteins in a single cell, but it's so difficult to measure it in plasma or a body fluid? Well, because of the abundancy differences we have in a body fluid. 90% of the body fluid is contained by about 10 proteins. So they take over all the signals that we get into our mass spectrometer, so they suppress everything else. And that's the problem we have with body fluids. In a tissue or a cell, you have a better distribution of abundances, and that helps you when it comes to the detection later on. we are working on different technologies to help us to get better in the body fluid section, also by depletion methods and so on. It's ongoing work on that side. So as a coincidence maybe, but today actually the paper was released that is just a result of another measure that we do. We do mRNA measurements, and this paper came out, and I won't go into the details. But this is in long COVID patients, and there was an indication of a signal disturbance in the JAK-STAT system, so as you see here, and a prolonged immune response in the patients. And this is the longest follow-up study, I think, that have been done on mRNA in long COVID patients. Out there for you to read if you want. So, complexity is extreme, as you know. We are struggling with finding out what's going on in the patients and seeing what we can do, what kind of treatments would be useful. So that made us think a bit about what to do next here, and we decided to launch a project that we call the BioQuest. It's a ME/CFS targeted biomarker project, and the co-PI is Danielle Meadows, who is our member of the Open Medicine Foundation family. So very much thanks to her, we are going to run this project. And we are also very happy for inputs from you, what you think would be relevant, if you think anything should be added or deleted from our plan. Since we are launching it really now, we hope to get a fast response back from you in that case. So the idea is that we will then target the idea of finding robust clinical biomarkers, objective diagnostic test for ME/CFS. And that sounds good. Of course, you know, it's not an easy task. But what we will do, we will screen already collected biomaterials that we have in our hands, where we have full information about sampling quality and everything about the patients. And we will start with about 1,000 samples, 1,000 individuals, where half of them will be ME/CFS patients. We will have 300 disease controls approximately, and we will have about 200 healthy controls, healthy donors. And as disease controls, we have been thinking a bit about what is most relevant. In the clinic, when you see a patient, of course you have to decide what is potentially the diagnosis of the patient, and you have some groups that are more difficult to differ between than others, and we have just listed a few of those that we think would be relevant to include in our disease control group. And also very much welcome to get more on this list if so. Important is, of course, that there should be available samples somewhere that we could use and tap into, and that the quality of those samples follow our standard operation procedures, because otherwise they would probably skew our analysis. But as you see, some of these are very symptom-like when it comes to certain specific symptoms at least, and I hope we will learn from the disease control group also a bit about differences and similarities in the mechanism behind ME/CFS then. Yes, what we will do, we will import and use all the clinical data we have, so the electronic health records, all the clinical labs that have been run over years in the patients, pathology labs. If we have scans, MRI, CT, or PET, that will be also included. We will do proteomics, large scale. We will mainly focus on mass spectrometry techniques, but we may apply also other technologies like Olink or Simoa or other types of methodologies. We will do metabolomics, and as I said, we will most do targeted and absolute quantitative metabolomics in our hands. They might be used for some screening methods also if you have certain families of molecules that we can't reach with our technology. We will do cytokines because we think that is, and the signals that we hear from other publications and in our own data show that cytokines are very important. It's something we need to target with selected techniques then. And then we will also do regular immunoassays for selected targets, like some of the markers mentioned here, BDNF, for instance, and SMPD, L3B, etc. And those can, of course, be expanded on. So in the end, we will end up with a lot of data, of course, and there we lean onto our bioinformatics groups to help us with evaluating the data, and also presentation just before me here is showing how much you can do, and Douglas also presented very well what kind of capacities we have in data analysis today with network analysis and AI-driven procedures, and I think that will help us a lot. But the end goal is not to end up with a pattern or something that is very complex. It should boil down to a few markers, hopefully, because that is what is realistic if you come to the clinical routine and robustness that a test would need to be responsive for. And then also the subtypes in ME/CFS will be targeted specifically to see if we can pan out what kind of subtypes that are. And I think definitely the data coming on the genetic side will help us in that future perspective also when it comes to subtyping. And then we come to the validation set and initial validation. The plan is that we will try to tap into this multi-center patients material that Vicky presented also. So we have a pending application there. We will do prospective validation studies. UK ME/CFS Biobank has, for instance, over 600 donors with nice collected material that we may ask to be using. And our collaboration with Akureyri Clinic will hopefully lead to a good biobank also that we can use. And since Fridbjörn, just by coincidence, also became the chair, I have to share a little visit to Iceland with you all. So Fridbjörn then, very nice fellow, as you know. He took me on a little trail ride on foot, not on horse, in a very beautiful area called Kjöllingafjölur. And dramatic landscape. The weather was pretty poor, but it was fantastic, right? We walked there. Definitely some challenges. 16 hours later, this was the news all over the world, unfortunately. My wife picked it up in Sweden, and so we had some issues. But we are both here today, Fridbjörn, and we are so happy to see each other every time. So it's a fantastic situation. So my take-home message for all of you, if you go into a biomarker hunt, if you go to Iceland also, bring a compass, because that is, it can be, you need leads and you need to have something to follow sometime. Otherwise you can end up in somewhat trouble. And by that, I just want to thank Open Medicine Foundation family then, that we are all working together across the globe, basically. All the CRC group leaders here, and Linda Tannenbaum, especially for her enormous energy and initiative to collect us and help us to run this forward. And to all of you also, all patients and all controls that actually now are giving us the opportunity to work in this field and also helping us so much with initiatives and also with supporting us with good ideas on what to target. So I think that is enormously important. And of course funding from Sweden, from here in UK, and also from other places in the world. So thank you so much for your interest.